Okay, yeah. So first of all, I would like to thank the organizers for the invitation and for giving me the opportunity to present some of our data here. So my lab is working on RNA methylation for the last couple of years, and today I'm actually focusing a little bit more about cancer and what we are doing to understand the function of the modifications in cancer. So I hope I don't have to introduce RNA modifications too much anymore. I just would like to point out that currently 171 modifications are known. It can occur on every nucleoside. And actually, I do like this review a lot because it points out exactly the two reasons why we are really interested in it. And the first one is the red ones. So over the last couple of years, all these red ones have been implicated in human diseases. So these can range from diabetes to neurodevelopmental diseases, mitochondrial disorders, or metabolic disorders to cancer. And the second reason why I find it interesting is the green highlighted ones that actually by RNA modifications really we gain such interest is technology development, because these are all the modifications we now can detect genome-wide or transcriptome-wide, and that obviously gives us the opportunity to understand the function in better detail. So this is a bit outdated, so we can do now a little bit more, but you can see that we're still only able to do that with a handful of modifications, so there's much more work to do. So when we started to be interested in RNA modifications, we basically focused first on the second step of gene expression, which is translation. But I also think that the basic translation machinery is very well suited to explain the importance of RNA modifications. So if you take, for instance, because the main components, ribosomal RNA, messenger RNAs, and the tRNAs are all three heavily modified. So if you take, for instance, the tRNA that comes here into the ribosome, the codon has to find the anticodon sequence, then you have to transfer the peptide on the peptide chain, and then the tRNA has to leave the ribosome in a timely manner. You can imagine that this is a highly dynamic process, and therefore I don't find it very surprising that tRNA is also the most modified RNA you find within a cell. So tRNA looks like this, and all these shaded positions here are sites that can be modified, not only by one, but by several modifications. I think what came out, which came quite, was apparent over the last 5 to 10 years, is that depending on the chemical nature of your modification and depending on where it sits in the RNA, it will have different functions. So for instance, in case of a tRNA, if the modification sits here in the wobble position, then it will be very likely be involved in codon optimization. If the modification sits outside of the wobble position, then it has been more linked to translation rates. So in particular, what I mean with this is there are several papers showing that these modifications are important of basically cleaving these tRNAs and producing these tRNA-derived fragments. And I will tell you a little bit about this in my talk. So that was first discovered by Frank Lücke, who found that the methylation site here, mediated by DNMT2, is important. We found something similar for the variable loop, which is mediated by enzyme 2, but also Christian Belodin showed, and this is slightly different, that with pseudouridine in this sitting here is actually more important than once you have generated these fragments to determine how they actually function in translation. So many years ago, when we got interested in RNA methylation in general, I think really it is important. The first tool you have to have a method that is transcriptome-wide and that can detect your modification at nucleoside resolution. So for methylation, this is actually one method is the RNA bisulfite sequencing, which is easy from principle, but very challenging for RNA because it is very difficult, I think almost impossible in my opinion, to detect it in unstable and rare RNAs like messenger RNAs, but it works very well for stable and non-coding RNAs, and tRNA obviously is one of that. So from the principle, it's really super straightforward. So it's based on the chemical conversion of unmodified cytosins to uracil, and if you carry this methyl group, you are protected from this conversion. So what you then have to do is you do sequencing, generate libraries, and then whenever you can detect your C, that meant that this C was methylated, and if you detect a T, that meant that C was non-methylated. So this is how it can look for tRNA when it works really nice. So this is an example that has been taken from patients that lack a functional enzyme 2 protein. So how you have to read this is that on this axis, so these are the different just example tRNAs, and here on this axis would be all the cytosins you find in your tRNA, and this is just the sequence reads. And blue means it was methylated, and yellow means it was non-methylated. So the things that are important to mention is probably if you look at the specific sites, they always occur at a pretty similar site in the tRNA, and this is the variable loop I just pointed out to you. The second important thing for the talk is that it is completely lost in the enzyme 2- patient cells, and that means it's a non-redundant function of the protein. What you should also notice here is the C38, which is in the anticodon loop, is not disappearing, and this one shouldn't disappear because that one is mediated by DNMT2. I think what I would like you to remember from this slide is that actually that this is not a rare modification. When we started off, the idea was that you methylate probably 1 or 2 tRNAs. That is not the case. We have done it in many, many different tissues and cells, and in a best case scenario, you can have up to 80% of all expressed tRNAs will carry this modification in the variable loop. So it's a very, very common modification. So, okay, so we now know where the modification sits, and we have confirmed that it's gone in the knockouts. The next question is obviously, well, what does it mean for the RNA itself? Why does it need the modification at this site? And the picture that emerged over the last couple of years is actually, again, I think quite straightforward, because if you think about it, all what RNA modifications in general do is, in my opinion, is they change the affinity to RNA binding proteins, right? And this is the same case for the tRNA. So here is the enzyme 2-mediated methylation in the variable loop. If the tRNAs lack this modification, they increase the affinity to an endonuclease. And in this case, the endonuclease is angiogenin. And angiogenin is really long known to cut tRNAs right through the middle here and generates these 2 tRNA-derived fragments that then accumulate in the cell. So you can measure these tRNA-derived fragments, for instance, obviously by northern blot and so on. We like to do it a little bit more genome-wide. We do small RNA sequencing, which is not without controversies. But I think if you do it right, you can make some assumptions about it. So for instance, if you directly compare the expressing and lacking tissues, you can see that you have a change of a specific tRNA fragments in the cell. So these tRNA fragments themselves, they gained a lot of attention in the literature, right? And they have probably many functions. And I think that is very difficult to determine because there will be, for sure, I think, also cell type specific. However, what we do know by doing many assays, for instance, by polysome profiling and ribosome profiling and so on, we do know that obviously they will inhibit global or de novo protein synthesis. And we do know that this happens during translation initiation, but also elongation, so on many levels. Okay, so what I would like to so the last step, obviously, goes very close to the heart of my lab, is we actually want to know, okay, now we know the pathway, but why is it important for cells, right? Why do the cells care to have methylated or unmethylated tRNAs? And here what we do is we use stem cells as a model system, and this is also giving you a very, with all the papers, the many papers we have done in the past, that actually these inhibition by these tRNA fragments or the low protein synthesis state is very, very important and actually functionally required for stem cells. And this is just show you how, an overview of how we did it. We like to use stem cells for that because they're actually very simple in the sense that they only have 2 decisions to make in their whole lifetime, right? They either have to divide and produce another stem cell, that is called self-renewal, or they commit to differentiation to basically generate all the lineages you need in the tissue. And we've mainly focused on the skin, but we also have worked a little bit in the brain. One thing that is important that we find that methylation is really not important for stem cells, and I'm talking about self-renewing stem cells. That is not just specific for methylation. I think it's safe to say that actually so for all RNA-modifying enzymes, they have been always shown, if you look at self-renewing cells, not to be important for the self-renewal process. They are always important to determine how the differentiation takes place and that you have the right lineages at the right time. So how do we know this? Basically, we know this because we knocked the enzymes out. And what you get is exactly this. You have like you do not affect stem cell renewal, but you do affect you delay basically the differentiation into the different tissues. So we have shown that in the skin and in the brain, and actually the worst affected is probably the testes because there the male mice are sterile and they don't produce any sperm. So this concept that the protein synthesis is really functionally required has since been also confirmed in the blood and in the brain by others. So I do think it's a very important mechanism. But what I would like to focus a little bit today on is what we stumbled across when we asked a little bit of a different question, and that was, so what does it actually mean for the cell metabolically when you change the tRNA methylation? So, and this started with simply the observation that enzyme 2 is like any other or most other RNA or DNA or even histone methyltransferases. They all use the same substrate for the methyl group, and this is SAM. So they take the group, methylate their C, and then they produce SAHA. And I was always wondering, because enzyme 2 is not ubiquitously expressed, it's very dynamically expressed, and you have certain conditions, so for instance in progenitor cells where it's very, very highly expressed. And I was wondering, if you take this enzyme out at this stage, what does it actually mean for the SEM metabolism, so the one-carbon metabolism? So what we did is to address this, we did a targeted metabolic screen by covering first the pathways that are closely related to the one-carbon metabolism, which is to first approximation, I would say, the methionine cycle and the folate cycle. So SEM would go in here. This is required for the methylation of DNA, RNA, and histones that produces SAHA. And obviously, these cycles are also very important for the synthesis of proteins, lipids, ROS production, and nucleotides. So what we did is, as I said, we did the metabolic screen, and what we used for this was first our patient cells. And this was basically, this is coming from, this is the mother here, so this is the cell line that is plus minus. And we had two children of hers, which are here. And we also compared this to a completely unrelated dermal fibroblast line we just purchased. And you can see here already that when we did the first unsupervised clustering, that this was really exciting for us, because this was sufficient to separate these according to genotype. And this, I have to say, was very tricky if you just do transcription analysis. So that was really, really exciting for us. But now you can say, okay, you have the patient cells, you have them in culture forever. We are talking about metabolites. No wonder that they're different, simply because it's, they are different sexes, they are different time, maybe different divisions, and so on. So to confirm that we really only look at the things that are specific to the methylation, we did also compare this to our mice, to our knockout mice, and we found that by mass spec and by NMR, we can, when we take the knockout skin compared to the expressing skin, that they're easily separated out just by genotype when we do this. So I find metabolic data extremely difficult to understand, because everything is related to everything. So how we started getting to the little bit more to understand what's going on, we first looked at the things that we expected to change, right? So in particular, the factors we found from the methionine cycle. So how I'm presenting this here is now the data is in red is pooled, both the patients, in gray is the mom, and they're all relative to the unrelated fibroblast line, right? So what you can see here is actually that we do find changes. And what occurred to us is that SAHA doesn't change, but SEM seems to be consistently increased. And that, in my naive explanation was simply, okay, you take an enzyme out that uses SEM, so no wonder that you probably have an enhancement of SEM as a metabolite in the cell. But then what actually led us to the first conclusion, that actually where I gave it away here, that what we are looking at is actually a cell that has been enforced into a catabolic state, came from these two metabolites here. This is symmetrical and asymmetrical dimethylarginine. And these two is basically, it's a very common modifications in histone and other proteins. But the beauty on these was that, at least to the current literature and to our knowledge, they only occur in the cells through protein degradation. There's no other way why they can be synthesized or how they can be synthesized. So that was leading us to the idea that basically what you have is you have a catabolic cell state. And that was kind of confirmed when we looked at other amino acids, that if they changed, I should say, then they were increased. The other consistent thing we found was that the TCA cycle, so the mitochondrial activity, was changed. So from here, I'm going to tell you a little bit more about mitochondria in a bit. From this, I cannot make any conclusions what actually is happening with the mitochondria, but we can say the mitochondrial activity was changed. So in hindsight, actually, I would say this is quite, we should probably have expected this, right? We have inhibition of protein synthesis, and now we are looking at a cell that is in a catabolic cell state. So that makes sense, because all it means is really that in the absence of enzyme 2, you degrade more proteins than you synthesize, right? But then we wondered, well, this must be, but nevertheless, it doesn't explain, because it's not a balance, because we shift towards the catabolic cell states. So how is this actually maintained, right? And to answer this question, what we did is we needed a system, actually, that where the very dynamic regulation of protein synthesis is important. So it is, for example, in development or any other condition the cell is experiencing. And obviously, one model system that came immediately into our mind was the response to oxidative stress, because during oxidative stress, this is exactly what happens, right? You have the inhibition of global de novo protein synthesis, but at the same time, you enforce gene-specific translation because the cells don't want to die. So what we did is we basically took our cells, and I'm showing you only some examples because this is published, where we basically took our cells and we exposed them to oxidative stress. So here, in this, and then we performed RNA sequencing. So what we did is, and I have to explain this a bit better because it usually causes some confusion. So obviously, many things happen when you expose your cells to oxidative stress. So what I'm going to show you is not the general changes. It's only the changes that are different in the minus versus the plus cells. So we are looking specifically at the things that are somehow dysregulated in the cells. So since we did it in a time course, and after 2 hours of the stress, we literally only find one GO category changed. And this GO category was all related to the mitochondria. It was mitochondrial translation termination, or it was the respiratory chain complex 1 assembly. So I think this is kind of really nice because it does confirm what we found to the metabolic screen. So we said, okay, but this is the first instance that happens. So what happens later then? So we did then compare at 4 hours. Now what you find is that many more gene oncologies come up, many more things change. But actually, I think if you look at this, it is always the same thing that changes. And that is, when you look closely, it is catabolic RNA processes and catabolic protein processes. So in a way, I really like that result because it really confirmed what we have shown on the metabolic screen. So instead of showing you all of the data that went into the data, I just wanted to summarize it. Actually, what does this mean in a more global sense for how RNA modifications work? And I think what we have learned from this is basically that you have a cell, and a cell obviously is in constant contact with its environment and has to constantly react and adapt its expression profile to the environment. And what we have focused on here was, as an environmental signal, was oxidative stress. So we do know already, I didn't show you the data, but we do know that cells exposed to oxidative stress, they downregulate very quickly NSAN2, and they lose the methylation on the tRNAs. So that then changes the tRNA fragment production in the cytoplasm. And then when you actually look, we have done several omics analysis, but if you really summarize all of these, what emerged was this scenario. So you have the tRNAs that get cleaved, the tRNA fragments inhibit global protein synthesis, but then this is reinforced by basically stabilizing the RNA and protein decay machinery. The second important step, what we learned through that, is that this goes hand in hand, though, with the mitochondria. And the mitochondria, instead of doing OXPHOS, they more tend to perform glycolysis. So what does it mean for the cell? It means for the cell that you turn from a very responsive cell to the environment to a cell that is, for a period of time, right, very unresponsive to the environment. And if you think of this, this makes sense if you look at oxidative stress, for instance, because oxidative stress causes a lot of DNA damage and also RNA damage. So you simply give the cell some time to decide, basically, should I repair the damage or should I just better apoptose? And this is basically kind of where we artificially, by taking the methylation out, where we put the cells for a prolonged time. So a cell that doesn't respond very well to its environment. And that's true not only for the stress I showed you. We have shown this, for instance, for also cytotoxic stressors like the chemotherapeutic agents in tumors. If I have time, I'll show you data a bit more detail later. But also you can also think of a stem cell, right, who's constantly exposed to cytokines and differentiation factors, right? So, but what I wanted today, when I wanted to show you some unpublished data, we actually questioned this. It's like, why is it actually? Why do the mitochondria activity come so reliably up in all our screens, and what does it actually mean? So we could have done, and to be fair, I have to also say that why we focused more on the mitochondria was actually one referee's fault, because this referee asked us in response to this paper, he or she said, well, your omics analysis is all nice, but you have to give me something that shows that your mitochondrial activity is really changed, right? And I said, it's fair enough. And we did it in a very crude way, I have to admit this, but I thought it was really giving some interesting results, because what we did do is we tried to do, during the stress response to oxidative stress, in the same cell, measure how protein synthesis changes and how the mitochondrial activity changes. So basically how we did this is we used the OPPURE incorporation as a measurement of de novo protein synthesis. This is very simple. What you do is, it's basically a OP-pure is a tRNA analog that is modified in such a way that you can make it fluorescent in the cell by a simple click reaction. This is obviously also intercalated in the growing peptide chain, and then you basically can have a quantitative measure at a certain time point how much protein is synthesized in the cell. And then you do this by FACS sorting. So what we did is we did use the expressing, enzyme expressing and lacking cells. And this is just showing you a typical response to the oxidative stress. Cyclohexamide obviously is a positive control because this is known to inhibit translation. But I think what we always found is that you have a really massive drop after 2 hours after arsenic incubation at this time. This is always recovering between 2 and 4 hours. The protein synthesis goes back, and then it kind of back into its normal background level. So you also find the same dynamics in the minus cells, but I would like to point out what we really showed is that here, what happens is that the cells start with a lower protein synthesis, and the amplitude to which they can react to the stimulus is much closer together, right? So they're basically a bit lazy in that sense. But as I said, I wanted to show you more of what the mitochondria do in the same cells, and for this we simply used the mitotracker, which measures the mitochondrial activity. And what we found was that it also changes in response to oxidative stress, which is probably not so surprising. But now what I would like to point out is we are looking at the same cells, right? So it does not, what you basically can see that is in response to stress, you have the activity. At the same time, where the protein synthesis goes down, is going up. And then it is basically also coming back to background level. So this was completely missing in the enzyme 2 minus. So they did not react. The mitochondria were very kind of resistant to react to do anything to the stress. So this basically caught our attention. It's like, why is this, right? Because it also looked to me that actually mitochondria are first, right? The mitochondria seem to react first to the stress, and then you have the cytoplasmic reaction of the protein translation. And as the best of my knowledge is, it's really not well understood how mitochondrial translation and cytoplasmic translation is actually coordinated in the cell, and there should be, right? So to answer this question, we could have looked, actually, I said also we could have kept to the enzyme 2 minus cells, because now we have published that they also methylate a couple of tRNAs in the mitochondria. However, we said, why are we not turning to the mitochondria themselves? Because the mitochondria do have their very own RNA methyltransferases, and this is enzyme 3. Enzyme 3 in this case is a tRNA again. In this case, enzyme 3 methylates the anticodon loop here, which in this methyl group is actually is required for subsequent modifications through oxidation to a formal cytosine here. And this formal cytosine changes the codon decoding activity of this tRNA. So the principle is actually really simple. An unmodified tRNA can decode 1 anticodon. A modified tRNA can decode 3 codons because it can base pair with other codons. So, and you can imagine if you now have a tRNA that can not only decode 1 but 3, that this obviously enhances the protein translation in general. So, what do we know about? What did we know about the enzyme 3? We did show actually that it was we stumbled across because it is. Loss of function was found in patients with mitochondrial disorders. It's really an exclusively expressed protein in the mitochondria. We did identify by bisulfite sequencing, so these are the mitochondrial tRNAs. Actually, it did only methylate, in this case, 1 tRNA, which was the methionine. Here at the variable loop, you can see that is methylated. It's not methylated in the patients, and it can be reinstated by overexpressing the wild-type protein. When you look at looking at the protein translation in the mitochondria is straightforward because there are not that many. When we looked at that, we could see that they are really all reduced, but if you reinstate the enzyme 3, they're coming back. So it's a clear effect on protein translation in this case. So, but we were wondering, okay, when we now want to know what is the function of the tRNA methylation in this case, again, I think what is really important for RNA modifications, you need to have a dynamic system where you can analyze the function. You need to find a situation where the modifications are important, right? And so again, we were wondering if really the mitochondria are important to instate a cell state with a low energy demand, then we need a system that goes through changes of this, right? And in this case, the system that came to our mind was basically the whole process of tumorigenesis, right? Because you have, in all stages, from initiation, growth, survival, and metastasis, the mitochondria have been implicated to be important for it, right? And that was mainly because of their, basically the relation probably also to oxidative stress responses. So we said, okay, that's a brilliant system. We just look through all of these stages and see where enzyme 3 or tRNA methylation in this case is important. So the model we chose was OSCC, which oral esophageal squamous cell carcinoma, and we needed basically a model to do this. And there we started a collaboration with Salvador, who just at the CRG, who had just established an orthotopic transplantation of oral OSCCs under the tongue of the mice. So it's an orthotopic transplantation assay. And we also know that these basically then metastasize into the lymph nodes or occasionally also into the lung. So what we did exactly this assay. We took cells that were expressing or lacking enzyme 3, and then we did the analysis. So what we did start off with was basically first, we used like an SCC cell line you really can buy from ATCs here. I mean, it's an established cell line that can be, that was known to metastasize. We transplanted these. You can see here's the primary tumor, and then we can get the lung metastasis. This was all fine. But we also wanted to already include some more patient material. So we used the patient-derived cell line, which was is called VDH15. And this is a very aggressive cell line. And it's also, you can see that they also metastasize into the lung. So what we did is we knocked it down, enzyme 3, with 2 different shRNAs. And what we did find in the SCCs was that you still get the primary tumor. I mean, overall, it probably looks a little bit that the tumor is a bit smaller, but to be honest, from all what I see from the histology, it doesn't look that much different. But what is very exciting for us is that they basically completely fail to generate the metastasis. And that was to a large extent also true for the very aggressive ones, that we did not find the metastasis into the lymph nodes, and we very rarely found a metastasis in the lung with the VDH. And that's just a quantification of that. So now we were wondering, sorry, I'll do this first. So now we are wondering, okay, what is the difference of these tumors? So why are they not able to metastasize in the cell, in the mice? And for this, we just did a transcriptome analysis. So we basically, again, took out tumors with expressed or didn't express enzyme 3. And then, obviously, we had to use the primary tumor because we rarely did get metastasis. And then we performed FACS analysis for GFP because we only wanted the human cells, right? And they had also express they would also express GFP. So these are GFP isolated then cells. We performed the transcriptome analysis. And this is just to show you that it really worked for the VDH. All I'm showing you now is for the patient cells. Worked very nicely in the sense that we got an okay difference between the control, the infected cells, and one of the RNAi. So you find differences. And if you do our 2 biological replicates, these are 2 different shRNAs. They are very, very similar to each other. So at first we said, okay, but we are interested to really know what happens to the mitochondria, right? So how we can use this data to find this out? And we did, for a moment, just forget about the significant changes. We did a little bit different approach. We just took some databases and asked, what are all the genes that we know that are involved in mitochondrial biogenesis or in regulating the mitochondrial functions? So there are, what we found is 781. I think there should be a couple of more, so we have to probably optimize this a little bit. But in our case, we found 781 human proteins, right? And we simply asked, how do these change in our tumors? And what we found was that if you compare, again, our 2 biological replicates, this is the 2 different shRNAs, they do correlate really nicely with each other. And these are basically all the ones we found covered on our transcriptome assay, and these were about 300. And you can see that most of them, actually all of them, change, and the majority of these are also statistically significant. So, but what it does say is we're really looking at a situation where the mitochondrial activity is clearly different, right? But this doesn't explain exactly how or why, right? So what we then said is, okay, can we learn something if you look a little bit closer into the mitochondria? So we just took now the transcripts or the genes that, according to their spatial distribution in the mitochondria, and asked what happens to them on our assay. So here we looked, for instance, for the RNAs that are located in the outer membrane or intermembrane of the mitochondria, the inner membrane, or generally integral to the membrane, or to the matrix. And here what you see, this is just a log fold change to the control, is that obviously most of them should be around 0. That was really obvious was the inner membrane was specifically repressed in the mitochondria. And we said, okay, again, this is very interesting, but why do you affect specifically just this population of RNAs? And then what we went back actually to our transcriptome analysis and asked, okay, now let's look at the significant changes. What happens in these? And when we did now go back to our tumors and just are very significant changes in the tumors themselves, and if you perform GO analysis, what you find is actually, again, these are really the most significant categories you find. So it's not somehow depicted. And it all comes back basically to the mitochondria. So you have this one structural constituent of the ribosome is mitochondrial related and also ribosomal protein, both ribosomal and mitochondrial related proteins. They're all downregulated, which is nice, which fits. But then these two obviously I think this is almost insanely significant, is basically again the complex one of the mitochondria. So these we find exactly the same, depicting what is happening here. So I find this is obviously, we are now having to show why is specifically the complex one affected. But I do think if you come back to the question, it's quite amazing that the metabolic reprogramming during tumor metastasis is basically strictly dependent on the efficient decoding of one mitochondrial tRNA, and that this is methylated. So does this mean that, does this mean, okay, we can just probably use this to inhibit metastasis in a patient or inhibit tumor growth in the patients? And I think with this, we're thinking about this quite a bit, but I think, yes, you can, but you really have to be considering that we're looking at a dynamic process. You're looking at a process of a tumor cell that requires OXPHOS or glycolysis in kind of waves, right? So you need to really consider as well that you force the cell into a low energy cell. But if you remember probably from the beginning of it, what I told you, another cell that is also requiring very low energy demands is a stem cell, right? Or a tumor-initiating cell. So you basically have to kind of depict what the functional role is. So in the last couple of minutes, I would like to illustrate you a little bit of what I mean by that by focusing on squamous cell cancer, which is also a cancer I like to focus on. It is also, does play quite a bit of a role, but I think just from the tumor point of view, I'm very interested in it because usually they're very well differentiated. And what this means, if you're interested in stem cell differentiation into different lineages, it obviously helps if you still see your differentiated lineages in a tumor. And the advantage also is that basically how stem cell differentiation occurs in these tumors is very well defined. So you're looking now through here, you have in the basal layer, tightly attached to the base membrane, are all undifferentiated cells. These are not all stem cells, but they are all undifferentiated. Interspersed into these are the actual stem cells, but the important thing is if you can basically isolate them by surface markers, but as soon as the cell leaves the basal layer, it will commit to differentiate, so there's nowhere back. So once you are out of here, you will undergo terminal differentiation into spinous, granular, and then stratum corneum, which would be the dead squamous here in the middle. So when we did, this is now going back to work we did quite a while ago, because when we asked, does enzyme 2 play a role in the tumor formation, what we found is very early on, and we used a skin model, which is here called the K5-SOS model, which is basically just constitutive expression of RAS in the skin, which causes these tumors for whatever reasons, only in the tail. We found that these are really, in the absence of enzyme 2, much worse. So they appear earlier, they grow faster, and they kill the animal much quicker. And then obviously we ask, does it have to do with the accumulation or enhancement of a population of tumor-initiating cells? So as I told you, you can isolate these tumor-initiating cells that have been shown by Blaine Fuchs and Lara Malanchi by using these two self-as markers, CD34 and interleukin alpha-6, and this is the population we focus on. And you can see already here that you have about a 4-fold increase in these in the positive cells, and that is this is just the quantification, and this really goes at an expense of the marker negative one, which are the terminally differentiated cells. So you have more tumor-initiating cells and less differentiated cells. And we obviously also showed that these are functional because you can transplant them and they will form the original tumor. But the question was, okay, so I've told you a lot about that these pathways are really important in the response to stress, right? So we were wondering at this time already, so how does it fit? How can you have an enhancement of this population, but then at the same time, these cells really struggle with the oxidative, with the stress response in general? So we asked, are these tumor-initiating cells more or less responsive to stress? And I mean, if you're talking about the tumor cell, you obviously ask whether chemotherapeutic agents, which would be the stress factor, how they react to that. So we did an assay where we tested this out. I just wanted to remind you, this is how it looks. You have the normal cells that do develop these tumors on the tail with 100% penance-free and very rapidly. In the absence of enzyme 2, they come earlier, and they grow much faster. So to test our hypothesis, what we did is we basically chose 5-FU as a chemotherapeutic drug agent, because most patients still receive this as a topical application of a cream for their tumors. And you can basically buy the cream in the pharmacy. That's what we did. And we treated the mouse tails with the same agent. And if you do this in the wild-type mice, you can see that, yeah, you have probably a little bit reduction in the tumor growth. You also get necrosis. But I was actually surprised to see how much they still grow. Now, on the other hand, if you do this in the background where they don't have enzyme 2, we completely, from day one, we basically abolished the tumor growth. And this is just to show you that this is not only in one, but in many tumors the case. So this is the most important one. So these are the tails that received, that have no enzyme 2 and received the 5-FU. One important thing is when you do these analysis, where you look at the stem cell or cell behavior in general, you always get the question, which is fair enough, this is all coincidental, right? You have no idea whether your tRNA fragments play any important role in that. So what we routinely do is, and we try to rescue the whole phenotype, but because we do know how the pathway works, and this is just to remind you that in this case, enzyme 2 loss increases the affinity to angiogenin, produces these tRNA fragments, and this inhibits translation. We do have angiogenin inhibitor that we know work very well because they do not, they basically protect from these tRNA cleavage, but we also know that these naked, so unmethylated tRNAs, they can act in translation really normally, at least to what we can say for now. So if you come over this, you have actually restored the translation. So we did a double treatment of this tumor genesis experiment, and so we just alternated, and this is now at the end. This is like a kind of double chemotherapeutic treatment, which I think also is obviously more toxic, not only to patients, also to mice. So we couldn't actually measure at the end the tumor size where there was a difference, but we could measure whether we have an effect on the tumor-initiating cells, right? If you remember, they are positive for CD34 and interleukin alpha-6, so we just isolated these cells, and we were measuring whether we have a difference. So this is, again, how the FACS looks like. So the population we are talking about is here, marker-positive one. And if you focus first on the marker-positive situation, and if in the enzyme 2 negatives one that have received the 5-FU, you can see that this population goes down. If you receive FU and the angiogenin inhibitor to rescue this, not fully, but to rescue the effect. And if you look at the marker-negative population, you have exactly the opposite, right? So I think what these experiments then tell you that is that in this case, the fragmentation of the tRNAs or the tRNA fragments, they do play an important role in basically in the response to the chemotherapeutic agent. So that was a while ago, and I would like to really raise your attention to there are many papers coming out that place RNA modifications into the cancer field. You have this M6A is the most common modification in the mRNAs, and then we have also, this was Pierre Close's work mainly, another modification in the tRNAs at the anticodon loop, and that was our study I talked to you about. But basically what you can see, you can, from colon to brain to blood to breast cancer and to skin tumors, you have really a wide range where these are important. I think in a way, trying to make this a little bit more, what do we like globally from that? I think if I would make prediction, is like the mRNA modification, if you mess up the mRNA modifications, you mainly affect like tumor development and tumor growth. These modifications in the tRNA are very often linked to chemotherapy resistance, right? So this is and metastasis as well. So this is coming back to this. So I think the tRNA modification will probably be more important in resistance metastasis, and these are in tumor growth. If I have a couple of minutes left, I would just like to summarize what does it all mean. So I would like to really, from all the work we have done, we really come back to the point that when you look at a stem cell or a tumor-initiating cell, that this is a cell that really functionally requires low protein synthesis. And we do know this because enzyme 2 is not expressed in the stem cells, but we do also know that it is dynamically regulated and highly upregulated in the progenitors, and later in the terminal differentiated cells is gone again. So what does it mean? For the tRNA fragmentation, it basically means that you have in the progenitors, the tRNAs are protected from these cleavage, and then they can act in protein synthesis. And, well, I wouldn't say they have to enhance, but they have to stabilize the protein synthesis, right? Now, when you look at the enzyme 2 knockouts, then you have the accumulation of these fragments, the inhibition of de novo protein synthesis, and morphologically what it means, I showed it with the example of the skin tumors, you have actually a physiological enrichment of these tumors in tumor cells in their niche. Again, it's very important to mention that if you look at the modifications, it is not a static system. You need the modifications dynamically. So this is the best stem cell is useless if it can't be activated to do its job, right? And this is exactly where then the methylation is needed to enter the progenitor cells. So, but that's not it. I would like to also, what we did show is that on a positive note for cancer therapies potentially is that these cells are stem cells, but they're extremely sensitive to oxidative stress because they basically lack the ability to dynamically adapt their protein synthesis and the metabolic requirements that go with it to the stress situation. So I do think that could be targeted somehow, I think. Because these cells rather undergo cell death than contribute to the tumor. So that was it from me. So we just moved to the decaf set, and this is the other brave souls that joined me. But so basically what I showed, the mitochondrial work was done by Sylvain, and the metabolic data were all done by Nicole. I also would like to really thank my former home and funder, Hansa Research UK, and my new funder, the DKF set. I thank you for your attention, and I hope you have some data, some questions.